I'm really thrilled to be here, and thank you to the EpIC founders and organizers, also with SGP, for really establishing this wonderful foundation for sharing our work on an international scale. So today, what I'm going to be sharing with you is some of our work over the past few years, where we're looking at really the allostery of ionotropic glutamate receptors, and in particularly the AMPA subtype, and then also some of our more recent work where we're looking at the glutamate gating mechanism. So I'd like to start with really the stars of this PowerPoint. The first work that I'll be talking about was led by Dr. Dylan Hale, and he's been working on our studies where we're trying to understand the allosteric mechanisms of how AMPA receptors are regulated. And he is a joint postdoc with Rick Huganier, and he's currently on the job market. And then also Dr. Anish Kumar Mandal, who's another postdoc in the lab, and he has been looking at how AMPA receptors gate in response to glutamate. And we, beyond our collaboration with Rick, we're very fortunate to have some very collaborative colleagues here at Hopkins. So we've collaborated with Albert Lau here, who has been helping us look at the molecular dynamics of allostery, and then also Dr. Vasanthi Jayaraman and her postdoc Lisa and her student Ulysses on these allosteric mechanisms. These are our funding sources, and we're very thankful for the Beckman Foundation for funding all aspects of our cryo-EM center at Hopkins. So today I'm going to be talking about our work on AMPA receptors, and why in particular are we interested in these. And if we break down our brain, we have roughly around 100 billion or so neurons. And all of our neurons communicate with each other by receiving input signals and then also giving output signals. And these the output signals are received by these dendrites on an adjacent neuron, and we call these communication junctions synapses. And there's roughly 100 trillion synapses in our brain, and most of these are glutamatergic, meaning that from the presynaptic neuron you have release of glutamate, and then in the postsynaptic neuron we're having this glutamate being received. And the receiving is mostly done by, as Elena mentioned, our favorite type of receptor, which are these ionotropic glutamate receptors. So these are specialized ligand-gated ion channels that are existing within this postsynaptic membrane. And there's various different flavors of them, such as the AMPA receptor, which we'll be talking about, and the NMDA receptor, for example. And when AMPA receptors first bind glutamate, they very rapidly are going to initiate this depolarization in the postsynaptic space. And this actually then contributes to the downstream activation of the other IGLR subtypes, such as the NMDA receptor, for example. And then if we could build up enough of this depolarization, then what we can do is then initiate a signal transduction response in the downstream neuron. And we're really interested in these from a basic sense, not only for how do a lot of these neurons communicate with each other, but also from a disease perspective. Because whether directly or indirectly, there are a significant amount of neurological disorders and diseases that can be linked back to AMPA receptors. And something that confounds a lot of this is that currently there's only a single drug that is approved for targeting AMPA receptors. So for us, we really want to use a structure-based approach to really understand mechanistically how these receptors function, but then also being able to leverage how these their structures in order to design new drugs. So just to give you a quick overall look at the receptor topology. So this is from one of our recent cryo-EM structures of a GluA2 receptor. It's just generally speaking, all AMPA receptors have this conserved three-level topology, where the fourth level would be the cytosolic domain under the transmembrane domain, which we've excluded from this figure. So at the top, of these tetrameric ligand-gated ion channels, we have the ATD, or the amino terminal domain. And this is really especially critical for receptor assembly, and more recently has been shown to be critical for pH sensing of the receptor by the Greger lab at MRC. Right below the amino terminal domain, we have the ligand binding domain, which is, of course, where glutamate binds. And the ligand binding domain is directly tethered down below to the transmembrane domain, which is going to house the ion channel in the center. And if we break down one of these domains, what we can see is typically in the AMPA receptor, we have domain swap, where we have the top of the ATD here. You can see this B subunit position is actually swaps over from a dimer pair, where up top here, it's paired with the A position subunit. It then swaps down here to then on the opposite face of the receptor to have a local dimer pair now with the C subunit. And we think this is important for actually kind of relaying conformational changes between subunits. And then if we break this down to look at a single protomer, we have the amino terminal domain here that leads down into the ligand binding domain. The ligand binding domain is coded into two stretches, so it's actually interrupted by the transmembrane domain. It comes back up to code for the second stretch of the ligand binding domain before finally coming down and completing the transmembrane domain with its fourth transmembrane helix. And then collectively, what this does in the ligand binding domain is that it breaks it up into two halves that we call D1 and D2 that kind of create this clamshell-like structure. And now if we break this down kind of into a really more basic sense, is that in a sense, gating is really just coupled to the channel below by these LBD TMD linkers. And if we kind of now really just break this down and we look at two subunits of the tetramer, what's kind of really critical just in terms of gating is this D1 and D2, which are the upper and lower halves of the ligand binding domains. And this is where glutamate binds. And then we know up top here, we have the gate right below the ligand binding domain. And this was initially shown by Sasha Sobolevsky and Eric Wu's lab in 2009 with the first overall crystal structure of the AMPA receptor, where the M3 helices are really what's defining the ion channel, especially the upper gate. And then we have this reentrant loop and the helix, so the M2 helix here, and the loop beyond that really are going to be dictating the selectivity filter of the channel. And how I kind of like to think about and really just kind of simplify how AMPA receptors function is kind of thinking like AMPA receptors as like a Pac-Man gated ion channel or a kind of an evolved version of Pac-Man, where these ligand binding domains are trying to track down glutamate. Once they track down glutamate, they're closing down upon the glutamate or the cookie in the sense of Pac-Man. But then instead of actually trying to eat the ghosts around in solution, they're pairing up with their local dimers in the structure. And when they're paired up in these local dimers, they can then actually put tension on the ion channel helices below, which then allows cations to get into the postsynaptic space. So just to give an overall look at allostery and gating, we know that the receptors really occupy three primary functional states, where we have the resting state of the receptor here in cartoon. And then when we add glutamate to the system, we have this clamshell closure in this pre-activated state. And from here, the receptors can transition into either the active or the desensitized states. So the current understanding is that during activation, which has been in the context of cyclothiazide here, which is a positive allosteric modulator, we know that this is actually preventing desensitization and then holding the channel open. And then where you can see here, this motion of the D2 is swinging up and kind of pulling open these helices. And then down below here in the desensitized state, we think this occurs from when these ligand binding domains kind of in a sense are visually rolling away from each other. And the principal consequence of that is down here at these linkers, which are then kind of relaxed and then restoring the closed channel gate. So in addition to these positive allosteric modulators, what we also know is that there are these negative allosteric modulators. So there were foundational work from Mayer labs, Eyal Stern-Bach, and the Sobolevsky labs. They're really describing how these negative allosteric modulators bind kind of around this channel collar region in the receptor. And this is how negative allosteric modulators prevent gating to happen. But something that we are really quite interested in my lab was that one, from a structural point of view, we didn't understand how negative allosteric modulators work structurally in the context of glutamate when we're having glutamate within the system. And something else that we wanted to know is that if you have a negative allosteric modulator that binds in this binding site, but you also have a positive allosteric modulator that binds at a completely distinct binding site, can these two compete for control of the AMPA receptor? So what we began looking at about a year and a half ago was how do negative allosteric modulators work? And then can positive and negative modulators compete to control function? So that's going to be some of the first part of the seminar that Dylan Hale tackled. And then also more recently, something we've been wondering is that do the positive allosteric states, which are currently encompass all of the published activated states of AMPA receptors, do these represent the glutamate activated state? So a receptor activated in the complete absence of any sort of modulator. So to get at this first, what we thought about doing and what Dylan ended up coming up with were these kind of two modes for looking at this in the context of cryo-EM, where one we would purify our AMPA receptors in complex with a auxiliary subunit TARP gamma 2, which positively enhances AMPA receptor activation in situ. So in mode 1, where we would first incubate this with cyclophiazide, and then immediately before making our cryo-EM grids, we would spike in glutamate along with Geikie 52466, or Geikie as I call it in this seminar, which is a prototypical negative allosteric modulator for these receptors that bind along the collar region that I showed earlier. And then the second mode, what we would do is we would pre-incubate both cyclophiazide and Geikie, and then right before we make our grid, spike in glutamate. So generally speaking, these are how the cryo-EM grids would look. And then what we get from both conditions are something that looks like this. So we have this overall receptor that resembles kind of the typical Y-shaped arrangement of an AMPA receptor, as you can see here. We have 4 TARP gamma 2 subunits. I'm kind of decorating around the core of the receptor. And then we have glutamate that is bound in each ligand binding domain. We have our negative allosteric modulator that's bound around the ion channel collar region. And then what was missing is a cyclophiazide. But before I get into that, we'll talk about kind of some of the key features of the transmembrane domain itself. So technically, what we did to get to high resolution down here in our in the transmembrane domain where the ion channel is, we computationally deleted all the signal from the entire protein with the exception of the transmembrane domain. And that enabled us to identify or see these lipids that are kind of really decorating the core of the transmembrane domain. And then up here, definitively define the closed ion channel gate, which has these 4 Geikie molecules bound around it. So if we rotate this 90 degrees, what we can see is this top gate of the ion channel here, which is quite constricted. And then you'll see here kind of looks like kind of like a Dorito is wedged in on each side of the ion channel, and that is the Geikie molecule. And so then in correspondence, what we see is a completely closed ion channel. So then what we got interested in is then what happened to basically cyclothiazide? And what we then looked at was this overall comparison here. So in white, what we have is the activated state of the receptors. So just in the presence of glutamate and cyclothiazide. And then in the colored subunits, so purple and orange here, are the this state from our cryo-EM. And what you can generally see is in the transmembrane domain, you have a general agreement, unless you're at the top of the channel here. But then the ligand binding domains here are completely different. And if you break this down into a singular subunit, what you can see is that in the activated state and our Geikie inhibited state, both the clamshells are maximally closed along the neurotransmitter glutamate, which activates the channel. And really, the primary difference here that we see is then Geikie is directly then interfering with kind of this hinging or kinking outward of the central ion channel helix. And then because of that, to accommodate what is happening down here, then you have a complete rearrangement of the ligand binding domain. So then if we break this up into local dimers in the ligand binding domain across the tetramer, we have local dimer pairs that we discussed earlier between the A and D subunits or the C and B subunits. And what we see in the inhibited state is that these ligand binding domains are kind of really up top at D1, really spreading apart from each other. And what's significant about that is that this puts the kind of what we would call the state of the receptor into a desensitized-like state, where typically in how we think about desensitization is actually decoupling of these D1 interfaces. And that's significant because in these PPLM bound, so that is a perampanel and perampanel-like molecules, which are in this class of negative allosteric modulators that Geikie is in, these have previously been characterized and studied in the absence of glutamate. So what we think in those previous studies is that these actually aren't inhibited states, but they are actually states where the inhibitor is bound to the resting state of the receptor, which would actually be more consistent with our current knowledge of AMPA receptor structures and function. And importantly, then what we see during this allosteric modulation in the presence of both Geikie and cyclothiazide is that we have a completely ruptured cyclothiazide binding site. So if we just do a direct comparison between the just cyclothiazide bound state in the to the Geikie cyclothiazide state, this state is completely ruptured, and which is then coupled with this D1 separation in these ligand binding domains. So then we think then that Geikie can directly basically compete and outcompete the positive allosteric mechanism from cyclothiazide. And then what we did to further investigate this is we collaborated with Vasanthi Jayaraman's lab down in Houston to do FRET, where what we were directly looking at is then coupling between these between the D1s and the ligand binding domains. And what we see also directly with single molecule FRET is decoupling of these ligand binding domains in the presence of Geikie. And then this gives us kind of this overall now view of how we are thinking of allosteric modulation, where in principle we have these ligand binding domains here that are the direct kind of gating machinery that is going to open up the ion channel. We have positive allosteric modulators that lock in the activated state. And so this basically allows tension to be placed on the M3 ion channel helices. But then negative allosteric modulators, which are wedging right around the, basically around that top gate of the ion channel. We believe these work by actually kind of forcing the receptor into a desensitized-like state and then blocking the active state transition. And then if in the context of allosteric competition, you can have both of these small molecules bind at completely disparate sites. But because of of really kind of this disparate effect where this negative allosteric modulators are kind of wedging around this central channel. This is able to actually wedge or kind of completely expel cyclothiazide from the binding site of the receptor. And then what we began to get interested in from just looking at allostery a lot is we began to realize, or we realized that all of the current activated states of AMPA receptors are with either cyclothiazide or other positive allosteric modulators. So here I have a few of the activated state structures in the literature of every flavor of AMPA receptor. And so what Anish began to think about in the lab was how does glutamate activate AMPA receptors in the absence of a positive allosteric modulator, which for us was a really fundamental question. So we've been working on this a lot in my lab with trying to capture this with time-resolved cryo-EM, because AMPA receptors activate on a very fast time scale. But we ultimately were unsuccessful with that, but we began to think about how and ask whether temperature can affect the activation of receptors. So kind of pointing to this was this 1992 study from the Boundary Lab where they used hot AMPA to then directly observe that as you increase the temperature in slices to just physiological like temperatures that you would expect in the brain, you have increased binding of AMPA to excitatory synapses. So what we thought was that perhaps we could just use physiological temperatures or just temperature in general as a tool to capture the activated state of the receptor in the absence of any positive allosteric modulator. So what Anish went ahead and did is we came up with this general scheme where we would take our purified receptors, we would incubate them at physiological temperatures, where at first what we ended up doing was actually doing this at 42 degrees because we were quite interested in some TBIs, for example, where you can have increased temperatures and how this can potentially affect basically AMPA receptor gating. And what we did at 42 degrees here is then we first put our receptor, incubated this for 10 minutes at 42 degrees. And then what we would do is then we would immediately spike in either glutamate or not add anything that's also already preset and pre-incubated at temperature. And this would then all be done in a Vitrobot from Thermo Fisher that is already preset to temperature. And then we would go ahead and vitrify and stop kind of this gating cycle. So to give you kind of an idea, these are how these single AMPA receptors look in ice. So as you're freezing these samples, it's a little bit challenging because you can imagine at 42 degrees, you have a lot more humidity, so there's a bit more ice contamination. And then what we can go ahead and see is that the 2D classes actually look quite nice, where they give us basically views as we would we're hoping to expect from the receptor. So then what we were able to do is kind of reconstruct a general gating ensemble from the snapshots that we were able to capture here. So from the APO data, what we see are two resting states. So these resting states are extremely similar. So the principal difference is just that the ligand binding domains are moving. There's some little bit of conformational heterogeneity between the ligand binding domains and the transmembrane domain. And then we also just completely delete out the amino-terminal domains. So we're able to kind of parse out the different functional states. And this enabled us to get to see one activated state in principle, and then two desensitized like states, which similarly to the resting states, or there's just some conformational heterogeneity between the ligand binding domain and how it moves relative to the transmembrane below. If you take a central slice through here, we can see all the main features of the ion channel gate. So up here, for example, in the resting state, we have the closed upper gate. We have the selectivity filter down here. In the activated state, we have an open gate up top, and we have a very defined selectivity filter down here. And then similarly in the or juxtaposed to this in our desensitized states, the upper gate begins to close. So then you may notice we did this a little bit backwards initially, where for us we had a lot of microscope time available. So what Anish did was he just came up with this scheme right here when we had some extra purified protein and threw it on the grid. So then what we want to go ahead and look at is this actually true where physiological temperatures or this 42-degree temperature is allowing us to kind of capture this activated state where in principle we shouldn't have been able to do this without positive allosteric modulators. So what we went ahead and did is we went down to Houston with Fasanti again and her great postdoc Lisa, and we started to do then single channel recordings where we are then looking at glutamate responses at various temperatures. So typically here in my lab, we would usually make our cryo-EM grids, for example, at room temperature, which is roughly 25 or so. And then as you increase temperature to 30, 37, and 42, just even very quite qualitatively, we're beginning to get many more openings. And then also, higher conductances as you would probably expect, because as you're increasing temperature, you're getting more ions going through the channel. And what we began to notice is then, although the open dwell time was beginning to decrease, as you might expect, because you're having greater transitions, the open probability of the channel at 42, for example, is around 30%. At 37 degrees was around 20 or so percent with our cryo-EM construct. And then typically where we would be making grids at this temperature, it was really quite low open probability. So what we began to then see, and that we can see in the supplement of this preprint, is that although the open dwell time was decreasing a little bit, the resting time or the resting dwell time of the receptor was really increasing or decreasing even more dramatically, which is then what actually enables kind of these higher probability states at physiological temperatures. So here's an overall look at the glutamate activated state of the receptor. So kind of similarly, which you'll probably be getting familiar with of this overall layout here is we have our ligand binding domains, our transmembrane domain below. Again, we deleted computationally our amino-terminal domain up top. We have 4 TARP gamma 2 or stargazer molecules kind of decorating the central channel. And then, of course, our ligand binding domains form these local dimers. And to actually look at this activation mechanism, what we can see up here where this channel is now being held open below by these ligand binding domains. These ligand binding domains local dimers are really held intact by a quite extensive salt bridge here, which we call the activation bridge. And this basically lines this entire top of this D1 and D1 interface between ligand binding domains. And we know from seminal work from the Guo and the Mayer labs that ablating these salt bridges had profound effects on really reducing the activated state dwell time, and we're kind of really forcing a desensitization. And then what was quite exciting for us that we'll look more closely at in this ion channel pore is that this is the most activated and kind of open channel of any glutamate or ionotropic glutamate receptor channel thus far, where kind of at this central selectivity filter area that was defined by us, and then more recently by the Student and Plested labs, is that we have very minimal constriction at the selectivity filter of around 2.5 angstrom or so. And then the upper gate of the channel up here is also wide open. So this is really quite exciting for us. When we looked more closely at this is, so what we do in terms of just conceptually is because of the symmetric pairs within the receptor, we break this down to the B and D pairs and the A and C pairs. So if we then look at the B and D pairs and just how these ligand binding domains are directly coupled to the M3 ion channel, what was really quite shocking to us was how the channel is opening and just the presence of glutamate. So we see that these channels directly open at this serine 615 kink or hinge. And this is really was quite exciting to us because this is happening right at the beginning of the Seitenlauf motif. And this is a motif that is conserved in every ionotropic glutamate receptor, and this wasn't observed before structurally. And so this is happening in the B and D subunits, and we have this entire Seitenlauf motif outlined in pink. And then this channel collar here, which is actually right where Geikie and the negative allosteric modulators bind, is actually we see in a Dillon's paper earlier this year, and what I was showing earlier, that the channel collar actually kind of integrates the entire Seitenlauf motif. So we think these negative modulators are so potent because the entire motif is actually necessary for opening the channel. In the A and C subunits, what we then see is then rather than a kinking or a hinging down at this serine 615, we actually see it at this pentultimate residue, so this alanine. So you can see this right here where these helices kind of begin to kind of unfold. And that's quite significant because this is the site of the Lurcher mutation that was discovered by Heinz, Linden and colleagues in the late 90s and studied for in the last few decades, where point mutations at this kink site or this Lurcher hinge site really cause spontaneous openings of the channel, and we think that's because really this is fully ingrained in the opening mechanism of the channel in the absence of positive modulators. And then how all these basically how all the tension is then directly coupled to these ion channels is through here, where we have this M3S2 or coupling with this ligand binding domain, which is basically the linker that's directly pulling open the ion channel. And this is done through this hydrophobic pocket. And this was pointed out really beautifully by some of the pre-activated state work from the Google lab in the mid 2010s. And then what we began to wonder really is then how all of this is like different to the positive allosteric states that are currently in the literature. So just to give you kind of a recap of how this looks across the scale of a tetrameric channel, is that when you pair this across all four subunits, you have the B and D subunits that are kinking or hinging at this first serine in this motif, and then the A and C subunits that are then hinging away from the central ion channel at this Lurcher kink or hinge. So all four ion channel helices in the glutamate activated state are actually hinging away from the ion channel to open it. So what we went ahead and did is we then just directly compared this to the activated state of the receptor, or the PAM activated state of the receptor here in purple. And what we can see here is this, what we call this activation bridge, as has been previously identified, is fully intact. And the major difference here, though, is that cyclothiazide is completely missing from its binding sites, as you would probably expect, right? Because it's not included in our condition. So here, where really cyclothiazide is kind of preventing the channel desensitization. And then how does this look overall? Where there's a very fine difference in the helix, in the pore helix itself, where in the glutamate activated state, we have this hinging at that first residue in the Seitenlauf motif. But this is actually different than what we see in the cyclothiazide bound states, where this hinging is actually being observed kind of further up with this M3 helix in the B and D subunits. And then in the A and C subunits in the positive allosteric modulator states, there's actually no cyclothiazide found, or sorry, no hinging there. And what this kind of couples to, when you couple this difference in the hinging across the receptor, is you have this maximum kind of opening here, which is quite similar in both states. But then because of the direct tethering of this ion channel helix M3 to the reentrant loop helix M2, you have a actually kind of dilation at the principal site of the QR site, or Q586, which really defines channel selectivity of permeation. So kind of what we're beginning to think is that because it's quite now well defined that cyclothiazide through binding right here is actually kind of preventing decoupling of the D1-D1 interface, which would actually enable desensitization to happen. Because that's happening, we think then down here in the ion channel, these positive allosteric states could actually be post-activation states that are beginning to refold the helix a few turns. And then what we then wanted to do, kind of away from allostery, is just use these snapshots that we have from cryo-EM to begin to estimate the gating ensemble, which is I would just like to make very clear that it is a gating estimate because these are all just snapshots that we get from cryo-EM, and we're trying to use these to approximate how gating happens. So here, what I have are the core AMP receptor structures from each of our principal states from cryo-EM, and where the TARP gamma 2 subunits are excluded just for simplicity's sake. And where in the resting states, they're just completely unligated. So they're in the apo states and activated. We have glutamate bound there, and similarly in the desensitized state. And what's really critical here is then if you kind of overlay all of these, the pore radius for all of these states, is that we could see here, which would be right above this central cavity and right above at the start of the Seitenlauf motif. You can kind of see here, even just qualitatively, how these pore radiuses are kind of hinging like this between the states. How the hinging actually initiates at that first serine motif and not at the alanine, which would actually be all the way up here in the pore plot. So how does this happen overall? So if we do an overlay of the entire receptor, kind of similarly to the Geikie diagrams or the inhibition diagrams I was showing from Dylan's work earlier, where the overall changes in the transmembrane domain are a bit minimal, other than kind of up in this channel gate, where the principal changes are in this ligand binding domain. So this is kind of an overlay of the overall receptor aligned directly to the M3 channel helices. If we break this down to a single ligand binding domain, what we see is that glutamate binding actually from the resting state here in blue to whether it's the desensitized or the activated state is causing a rough 26-degree kind of closure or upswing of that D2 to kind of really clasp down on the neurotransmitter. When we then begin to couple this motion with kind of the ion channel pore and thinking about this with these opposite ligand binding domains, what we see is that the B and D subunits, kind of this principal motion here, which would in this idealized case would come from a D2 upswing. The direction of the swing and kind of the distance from where it's being tethered is actually pore distal. And where in the A and C subunits, where the ligand binding domain is directly tethered to M3, is actually pore proximal, which you can visualize right here from the M3S2 linker. So then what we think is then that because of this kind of pore distal versus proximal, the B and D subunits actually have a kind of a greater pulling torque or a force, which enables a kind of a deeper kind of unfolding of that central M3 helix that we call the S hinge or the S kink. And then the A and C subunits don't exert a similar torque because of their proximity to the M3 helix, where then they only are kind of unhinging or unfolding to the lurcher site. And then similarly, when you get desensitization in the desensitized state, when you have these receptors kind of rolling back, you get refolding of these helices. And then if we break this down now to look at not opposite LBD dimers, but local LBD dimers, we can see basically in this activated state, compared to the resting state, the principal difference is really spreading apart of these of the D2D2 distance to really pull open the ion channel. And then in desensitization, which cyclothiazide would typically block, this is then kind of becoming relaxed to restore the channel gate. So if we rotate this 90 degrees and now couple our local dimer pairs across a local tetramer pair, what we end up seeing generally as we traverse from state to state is that from the activated state, we're getting a relative counterclockwise motion, which then expands this activation ring, which is kind of directly tethered below to the ion channel. And then as we go to the desensitized state, these have a actually kind of restore that ring to close it into a clockwise motion. And then similarly, going back to pore distal versus pore proximal, the B and D subunits do not move as much, but actually unfold that M3 helix to a greater extent. And we think that's because they're just pore distal in terms of their motion, as opposed to the A and C subunits, which have a much greater motion, but a less kind of effect on the hinge. And how this all looks kind of motion, kind of in motion through morphs is similar to this. As we activate with this motion, we're opening this this activation ring. As we desensitize, we're restoring it. And as we go from a desensitized to resting, we're kind of keeping that ring intact. And then in total, what this ultimately ends up giving is that these distal subunits are the ones that fully then unfold the helix or the pore helix to the S hinge. So right at the beginning of that motif, the Seitenlauf motif that is conserved in all glutamate receptors. And then the A and C subunits are unfolding these M3 helices down to that lurcher hinge. And then, of course, this gets restored in the desensitized state. So then just to kind of wrap all of this up to give you kind of an overview of how we're now thinking of these gating states versus the allostery states, where here I have the ATD eliminated just for simplicity, where we have these the clamshells of our ligand binding domains, and we have our B and D subunits, which are symmetrically the same, and kind of juxtaposed to their A and C subunit partners. We have our upper gate, and that's composed of the top of the M3 helices. And then something that I glanced over for time is that we think this QR site, which really defines the selectivity filter, could be a potential lower gate of the channel. As we add glutamate into the system, then we can go through these pre-active intermediates to transition between different states. So in the beginning of my talk, I went over negative allosteric modulator, so NAM inhibition, where these NAMs are putting the receptors into a distinct state that is desensitized like, where in desensitization you have these clamshells bound, but they roll away from each other in local dimer pairs to kind of keep this relaxed state of the ligand binding domain TMD linkers. In the activated state, we kind of have this subunit non-equivalence, where the motions of the B and D subunits are unfolding the helix down to this S kink or hinge at the beginning of the Seitenlauf motif. The A and C subunits are doing this to the lurcher kink or their lurcher hinge, which is further up on the M3 helix. And then in the PAM activated states, where you have cyclothiazide actually preventing this desensitized state, we think that the helices are beginning to actually relax a bit more, which enables refolding of the A and C subunit helices, and then a partial refolding of the B and D M3 helices to this alanine and the penultimate motif. Just some quick conclusions is that we think NAMs or negative allosteric modulators shift our receptors into desensitized state. That glutamate activation is distinct from PAM activation. So there could be minor differences there where it is possible, of course, that we have different subconductance states in these different receptor studies. We see that temperature, and in particular physiological temperatures, augment AMPA receptor function to the typical temperatures like room temperature, where a lot of us record from. And then in the glutamate activated state, we see that the gate is actually kinking or hinging in all four subunit helices. So the serine, that's the site and LAF motif in the B and D positions, and this Lurcher site in the A and C positions. And then the non-equivalence of basically the subunit positions relative to the physical gate itself is what kind of gives rise to these differences in the hinge. And thank you again.
Okay, so I'm going to read out the questions, and then Ed, you can answer one by one. So first one is from David Colquhoun. So the three states that you draw remind me of how Changeau describes the nicotinic receptor. But single channel measurements show that you need a lot more than three states to describe the function of ligand-gated channels. And then at the bottom, so it might be possible, so single channel recordings may not be possible with AMPA receptors, but then during the talk, we saw that they are definitely possible, difficult, but possible. So it might be possible to analyze single channels at high temperature. That is the question. Is it possible to analyze
that's a great
single channels at high temperature?
And so that's a fantastic question, about one thing, there's definitely more functional states, structurally that we can't currently reconstruct with cryo-EM. So these are just the ones that are the most populated snapshots here. So one thing, just to point out that I skipped over in the talk, but from the functional recordings. So of course there's multiple subconductance states of the receptor where there's been characterized for subconductance states in IGLRs and AMPA receptors, of course. But the principal subconductance states at 37 degrees or 42, for example, in the case I describe here, are the maximum conductance states of the receptor. So the lower conductance states are not populated nearly as much as the O3 and O4, for example.
So the next question is from anonymous attendee. Single channel recordings, again, are performed in an intact membrane where physiological conditions, including temperature, influence structural conformational changes. However, in cryo-EM, receptors are often studied in a solubilized form on the grid at cryogenic temperatures. How well the cryo-EM structures reflect the functional conformations observed in electrophysiological recordings, and are there any specific strategies to ensure that you capture physiologically relevant state with cryo-EM?
No, yeah, so that's a great question. So, what we'll say. So I didn't have in here, but so everything that we put on the cryo-EM grid, we can actually just directly test with bilayers. And so while these EPhys recordings were on intact membranes, we've done these exact same studies in the purified system, and the exact same thing happens with the cryo construct. I think in order to get to this directly, you would probably need subtomogram averaging on excitatory synapses. That are, of course, prepared and somehow high pressure frozen with temperature and preparing all of this. But I think, based on a lot of the immunogenesis and functional literature, this completely recapitulates the glutamate activated states.
I will read out a question now from one of the panelists, which is not in the chat. So it's from Tim Lina First, very nice talk, Ed, thank you. So the pore lining glutamines, right? Determine the larger pore radius at that level of the pore in the glutamate-only open state. Is it the glutamine main chains or glutamate side chains that are further apart in the glutamate-only open state than in the glutamate PAM open state? So how well are resolved these glutamines?
Yeah, so that's the glutamate, the glutamine side chains. And the local resolution of just around there, and the data I showed here is around high twos, low threes in or probably 2.8 to 3 in terms of the local resolution there of the channel. So it's not the best, but it's quite a good approximation.
Okay. So the next question is from May Weaver. Hi, beautiful talk. Do you have any insight into whether the differences you observe between the QR side of glutamate activated and PAM activated receptors may reflect a difference in ion selectivity in addition to changes in subconductors?
That's a great question. So that's something that we would have to do probably some additional MD and umbrella sampling between our pore differences structures, which we're currently doing. So you could imagine that with the differences in the pore radius, that could be better for like calcium versus sodium and partial dehydration or hydration. But that's a great question, but something we don't directly know yet. I mean, similar to the earlier question that was asked with the different subconductances.
So the next question is, have physiological PAMs and NAMs been described?
I'm sorry, physiological
Positive allosteric modulators and physiological negative allosteric modulators. Are there any physiological negative?
I see. Yeah, so in terms of physiological allosteric modulators, would be that something that we already have within the system, which are these auxiliary subunits, such as like TARP gamma 2, for example. And so all of these auxiliary subunits are biasing and kind of the different gating states of the receptor in still ways that are a bit complex and undescribed. And then I think what probably Andrew's getting at here is I would look at this preprint they put out that's looking at these differences in channel selectivity at the QR site.
So one last question, maybe there's more question in the chat, but we're running out of time. So the question is, how many channels were present in the single channel recordings where the effect of the temperature was studied? The open probabilities given are not very informative if the number of channels is not known. These open probabilities could still be very small at the level of single channels.
Yeah, so in the patches shown and then also in the preprints, these are single channels. So also it's quite a good estimation, I would say, in terms of the activation probability. So if you go into the preprint and you look at the activation probability at 42 degrees versus actually the cryo-EM reconstruction and all of the particles, it actually matches quite well. So somehow, whether it's through, let's say these positive feedbacks, like in the super activation that the Plisset lab has described, that kind of keeps these receptors in an activated state once you start activating them a lot. That kind of allows us to recapitulate the single channel recordings in cryo. We don't necessarily know, but I would say it's the single channel recordings in the paper are really well recapitulated in the cryo.
So I would like to thank you, Ed, once again for a fantastic talk and for this great data. And our next talk will be given by Ana Fernandez Marinho about ion channel biophysics.